Hello, welcome again for another discussion. So, I hope that you have enjoyed our previous discussion. Before we start, I want you to do this activity. So, I want you to get any sheet of paper, and I want you to list down at least three physical traits that you have inherited from your mother and another three from your father. So, I want you to write it in your paper. What do you think are the traits that you have inherited from your mother, and do you think are the traits that you have inherited from your father? So I want you to list it down on your paper, and you may pause this video, and then you can just turn it back or play it back on if you're done answering the activity. All right, thank you very much. Thank you for doing the activity. I hope you were able to reflect no, on those things. Now, what is the reason why did I let you to do this activity? What do you think would be our topic for today? So, today we will be dealing with heredity, genes, and all of those genetic materials because we will be dealing with genetic alteration. In this topic, we will be focusing on what are the things that we do to alter the genes of our organisms, and why do we do it? What are its advantage? And what are its disadvantage? How do this activity can affect environment in general? Okay? So before anything else, let me introduce to you our learning objectives. So first is to define what is genetic alteration. And the second one is I want you to determine the effects of genetic alteration in the environment. So these are again the things that we will be targeting or dealing with as we end the discussion or as we end this lesson. Now, let's talk about the concept of heredity. Genes actually, guys, was first described as an element of heredity. This was first observed by Gregor Mendel in 1865. So before Mendel actually discovered genes or the heredity concept, people during the ancient times before believe in theory of acquired characteristics. That means that if a particular parent who are not able to sing, okay, all right, oh, who don't know how to sing, would practice and learn how to sing, so what will happen? They would develop singing traits, and then they would pass it down through their offspring. Another example, if a particular parent is a dark-skinned individual, and then gipaputi nila, they believe that their child will inherit the white characteristics or the things that they have acquired before mating. So yun yung kanilang concept. However, that was being debunked by Gregor Mendel because of his observation, the very famous observation of pea plants. Um, his observation led him to conclude that specific traits from pea were passed on unchanged from a parent plant to the next generation. So there is a pattern when it comes to passing through traits according to Gregor Mendel. And then it was, patay na si Gregor. It was several years after Gregor's death, dara pa na hibawaan ang genetics or ang genes. So si Gregor, guys, wala siya nag-study about genes. However, he studied the pattern of trait or the pattern of passing of traits. So yon. Now, how do our traits pala or our genes um, can affect our phenotype or those physical characteristics or any characteristics that we have? Because again, this boils down to the coding sequence of our DNA. Okay? So yung lahat ng mga characteristics natin, behavioral and physical characteristics are being governed by our DNA. So how do our DNA do that? Now, as you notice, we have here the DNA. It would go to the particular protein, which is the ribosome. Okay? The ribosome, mo adto dito ang mga DNA sequence nato, and then the ribosome would synthesize amino acids or protein. Now, these proteins, guys, are being made based on the genetic sequence that we have. And even minor differences or changes to the sequence of the amino acid or protein would have a significant effect to our phenotype or physical or behavioral characteristics. So how do we do that? Or how do our system do that? So meron tayong ganito. For example, we have here, ito, katulad yan. Meron tayo, ito yung gene sequence natin. Now, the DNA, the first strand, would be attached into a ribosome, and then some tRNA would go here that would bring the together with its paired base pair na pwede nila ipares dariya sa unsa. And then as you notice, these different tRNA bring different proteins. Okay? Now, the different sequence here, ito ang mga sequence ng ating tRNA, nakadepende siya sa DNA strand ng ating DNA, yung coding sequence. Like for example, AUG. So, yung kanyang partner, that must be UAC. And then, these corresponding codons, guys, codons ang tawag natin dito sa mga tatlong ito. These corresponding codons do bring corresponding type of amino acids, itong mga circle. Now, the series of these amino acid creates protein. So, yung order or series ng ating amino acid is basically governed by our DNA kasi hindi siya or siya ang nag-i-influence kung anong codon ang talapit sa kanya with tRNA. That is how our genetic works. Kaya iba-iba tayo ng genes, iba-iba tayo ng genetic sequence, at iba-iba rin yung ating mga characteristics at phenotypes. Now, basically, in organisms, guys, we do have the same sequence actually with plants and other organisms. It's just that we differ with some very maliit lang with some sequence. Napakaliit lang yung differences natin with other organisms. Kasi isa lang na ating ninuno. Pero, yung mga different changes or different, yung very minor differences na yun can greatly affect our characteristics. Now, let us proceed. So, knowing how our genes can affect our phenotype, now discuss genetic alterations. Now, again, genes are passed from parents to offspring and contain the information needed to specify traits. These are arranged one after another in structures called chromosomes. So this is our chromosome, this is our DNA. The coil of this DNA will form a chromosome. A chromosome contains a single long DNA molecule and only a portion of which correspond to a single gene. Humans have approximately 20,000 genes arranged on their chromosomes. Now, what is genetic alteration? So we have two types of genetic alteration. The first one happened by controlled or selective breeding. What do you mean by controlled or selective breeding? Now, it is actually a genetic alteration from which human control the breeding of plants or animals. Example, if naakoy baboy na unuran, pero color black ang meat, undan siya, color black ang meat, i-mate na ko siya sa baboy na color white, pero payat. So, what would be my goal? Because I want to have a pig nga malaman nga color white. So, yun. Parang ganun. One example here is kana mga breeding sa dogs, right? Gusto na to nga taas siya og dalunggan pero mabalahibo. So, what we do is we select the parents who have corresponding traits nga desirable and then we mate them para mag-manifest ang kanyang traits sa iyahang offspring. For example, we have here, humans have changed the way dogs look and act by choosing which dogs to breed together. Again, the one in control here, or human is having control when it comes to breeding. For example, we have Mastiff. The Mastiff is known with a large and strong but slow and not aggressive. So, ito mga Mastiff. Ito yung behavior niya, yung phenotype niya. Large siya, strong, pero hindi siya slow at aggressive. Now, the Bulldog, on the other hand, is aggressive and quick pero small. Now, what happened? We breed the Mastiff and Bulldog and that creates the Bull Mastiff which has now a large, quick and strong and aggressive. And it is actually perfect for home protection. So that's what we call the selective breeding. Now, in genetic engineering, it is very molecular. Kung baka, we look into the molecular level of traits. So it is the method to very specifically cut out a gene from one organism and paste it to another. So what we do is we study the different sequence of the gene that would affect a particular phenotype or that would express to a particular phenotype. So i-study nato siya, i-cut out nato ang genes, i-dissect nato, and we look into the different sequence. So asa dapit diri ang maka-lead ani nga phenotype or ani na characteristics. So what will happen is we cut those characteristics and we paste it to another gene. So in that way, we can be able to combine the phenotype of the different organisms. So the first GMO was successfully engineered by Herbert Boyer and Stanley Cohen. They were able to combine the gene that encodes antibiotic resistance from one strain of bacteria into another. A year after that discovery, Rudolf Jenisch and Beatrice Mintz utilized similar procedures in animals, introducing foreign DNA into mouse embryos. So yun. So what happened is, for example, a particular bacteria. A particular bacteria is resistant to a particular pathogen. For example, ganun. So, for example, si bacteria A, nag-produce siya og kanang mga fluids that can kill bacteria B. Okay? So, ganun. So, what will happen now? That the DNA of that bacteria A will be transferred to another bacteria that would produce such kanang liquid. And then, ang katong liquid na gamiton to siya to be an antibiotic to kill another pathogenic bacteria. Okay? Another example. Um, for example, in producing insulin para sa sa tao, para iturok sa tao. Now, ang kanang mga cow, sila gyud atong main source of insulin before. Now, what happened is the genetic or the genes that we have nga nagaproduce sig insulin, gikuha to siya sa genetic sequence, gicut to siya by CRISPR, and gipaste siya sa bacteria. Now, what happened now? Ang bacteria na ang nagaproduce og insulin for medical purposes. And bacteria can efficiently produce insulin because they have smaller structure, they have simple structure. Gamay lang po gid ilang lifespan. So that means within their development, they produce the insulin. So mas paspas ang ato ang pag manufactured insulin. Even milk, guys, a lot of milk are being made by bacteria. So katong gene sa cow, kuhaon to i-cut and then i-paste to siya sa bacteria. Now what will happen? Instead nga ang cow na ang magbuhat og milk, it would be now the bacteria which will now create or manufacture the milk. Okay? So mas efficient siya. So yun yung mga benefits ng genetic engineering. Aside from that, ang ato ang mga plants. For example, ang ato ang plants kay dili siya or sensitive siya sa init o water. If too much heat, mamatay. If too much water, malata pod. So what will happen? What we do is that we cut the genes of that plant and then i-paste nato dito ang plant sa genes sa other plant in order to, again, to have another phenotype or characteristic. So, mahimo na ang plant og dili na kay siya mamatay sa init o dili na kay siya mamatay sa water. So, parang ganun. So, we alter the different sequence by cutting and pasting it to another organism. So, what are now its effect? But before that, genetic engineering technology is quickly advancing. Researchers have recently developed a new technology called CRISPR, which takes advantage of bacterial systems to simplify genetic editing, allowing for easier development of genetically engineered organisms. This technology could be used to expedite development of useful genetically engineered crops, facilitate disease elimination, cosmetic interventions, or even alter entire ecosystem. Now, the machine or the technology that we use in order to do such sophistication or sophisticated alteration of genetic sequence is what we call CRISPR. So, yun yung ating ginagamit. Okay. So dito na yung mga different development based on history. So in 300 BCE, una artificial selection muna ng wolves yung ginamit. And then we have all this evidence of artificially selected crops. So what do we do during these times, Kai? We still utilize selective breeding. So, yung mga good qualities of crops, ato siyang ginabreed with other crops. And then, sa animals naman, sa mga pet, ginabreed na to ang mga pet with good or desirable characteristics. And then, during 1973, that was the time that Boyer and Cohen developed genetic engineering. And si Jensen and Mitz, gidto nila gibuhat ang first genetic engineered na animal, which is the mouse in 1974. So, yun. US Supreme Court allows patenting, patenting of GE organisms for studies. Okay. And then FDA approves Humulin, the first pharmaceutical manufactured using genetic engineered technology. And then yun, yung mga tomatoes natin become GE na. And then approves the first insecticide producing crop. So yun, herbicide resistant na mga crops and etc. And during 2000 nga, golden rice engineered to mitigate vitamin A deficiency. So, naanatay rice nga naka-manufacture ng vitamin A. So, yun yung mga different positive effects of genetic engineering. Pero, what are its negative effects? The negative effects is biodiversity loss. An example of this is the expansion of GM herbicide-tolerant corn and soy, which are twined with herbicide, has destroyed much of habitat of the monarch butterfly in North America. So what happened? Once we introduce a genetically modified organism, which is a plant, man, or an animal in the wildlife habitat, what will happen? They become resistant sa mga pests. They become resistant with sa competition with regards to nutrients. So mahimo siyang um kumbaga mahimo siyang on top of the chain from which wala siya um wala siya mga kanang animals nga mag-consume sa iyaha kay resistant siya sa mga pest and then maka-survive gid siya. So what will happen? Magkaroon ng imbalance ang ecosystem. Mudaghan ang katong population atong mga genetically modified organisms and they would rule over a particular ecosystem. So what will happen? They would become or that would lead to ecological imbalance. Now, let's talk about pleiotropy. Okay. Pleiotropy, guys, that is when a particular genes will have a particular two phenotype that are not related to each other. So, this is actually a genetic problem from which a particular gene will impose health hazard. And then, for example, kani nga gene kay about sa heart ang iyahang about sa heart ang iyahang function. Now, or iyahang phenotype gina-express. Now, if nana siya defect nga gene, what will happen? The heart might have a problem. Pero, sa case of pleiotropy, aside from harming the heart, the genes would also harm the different organ system that are not related to heart. Another to that, aside pa nga ma-weaken ang heart, pwede po niya i-weaken ang bone. So the heart and the bone there are not related na mga phenotype, yet they are both affected by a particular or by one gene. That's what we call pleiotropy. So those are the different negative effects of GMO to our environment. Now, with a given problem, pros and cons when it comes to GMO and genetic alteration, what do you think now is the best thing to do? Should we embrace GMO or should we eradicate it and let nature decide what's best for it? So yun, that's the question now. That's a question. Or when would human would play gods? Because a lot of science research right now are pertaining to improving human health, improving human condition. So yun, para ma-extend ang life, makaproduce ng vitamins, and even guys, we already know how to engineer para sa offspring. For example, gusto ka nga imuhang anak kay puti, mestizo, taas og ilong. So you can change the sequence of that para sa imuhang anak. And it seems like we are now removing the way of nature of determining a particular trait of an organism. So how long would people play gods? And that's the question. So I hope you have enjoyed our discussion for today and you have learned something from me. Again, if you have questions, clarifications, you may message me and we will be answering all of your concerns and queries as long as we have time. Thank you very much, everyone, and enjoy the rest of the day.